So, hello, thank you for coming. This is the first in a sort of mini series of seminars that I'm going to be doing on machine learning, focusing on examples in biological sciences. So, as a quick overview of the things that I'm going to be talking about through the series, the first one, which is today, we're going to look at sort of linear regression as an easy starting point to sort of get warmed up into machine learning. And then throughout the rest of this series, I'm going to show a few different machine learning models, where they might be used, and also try and focus on some different types of input data that you might see. So looking, for example, in the third one at including sequences in your model inputs, in the last one at imaging data and how we might use machine learning for things like feature extraction, and also different, as I say, different types of machine learning model, where they might be used and what you might use them for. So the fourth one is looking more at interpretable models as well. So hopefully it will be an overview of a few different uses, and a few of the different considerations and challenges that you might have with different data sets and data types. For each of these, I'm going to be focusing broadly on one specific paper. They're not necessarily going to be journal clubs, though. I'm not going to go into great detail on all of the intricacies of the paper and their setup and findings. I'm more focusing on the models that they've used, the kind of processing that they've done to get the results that they get, and also hopefully show some practical examples of how we might go about building a similar kind of model ourselves, either on data from the paper or from sort of similar data types. So generally what I'm going to cover, and this is an overview for today, but probably for the other sessions too. So I'll do a quick introduction to machine learning as to what it actually is in comparison to more traditional experimental data analysis approaches. I'll do a quick introduction to the paper, go over the data sets used and what data is actually included, and then go through some of the pre-processing approaches around data tidying or feature extraction where that's needed, go through the main model that they use and sort of conceptually how it works. So I won't go into any of the sort of maths behind it really, but sort of thinking about how we can logically think about how the models work. And then look at how they validated it, so how well their model works, and hopefully go through and run a similar model in Tidymodels, which is what we like to use. And then finally, just some further exploration from the model results and some of the things that the papers did with the results from their models in the end. So a good place to start, I thought, is what is machine learning? So we've all heard about it, it's mentioned a lot, but what actually is it? And how does it compare to our usual sort of data analysis workflow? So in a usual data analysis workflow, we will go through collection, preparation, formalization, and we will get some kind of outcome from this. So we'll start off with some kind of raw data, whatever it is that we've collected. We'll then visualize and explore our data to see how it's behaving, if there's anything strange going on, if there's anything we might need to do to it, any filtering or cleaning that we might need to apply. And that is usually a cyclical process. So we might do multiple rounds of visualization and exploration, filtering and cleaning. This is also the case when we're doing machine learning. This is super important, whatever kind of data analysis you are doing and whatever the outcome you are aiming for. You always need to do some visualization, some exploration, and probably some filtering and cleaning to make your data how it needs to be. Where we then differ is how we formalize our results, and how the outcome that we might get from that. So if we think of sort of traditional statistics, so our t-tests and our ANOVAs and things like that, that's our way of formalizing our data, formalizing our results. which will then give us a statistical result that we can report. So we can report our p-value and our test statistics and things like that. And that will give us our outcome because we can say if our groups are statistically significantly different or not, or whatever it is that we're looking at. If we're doing machine learning, we will, instead of doing sort of base statistics, we will do some kind of modeling on our cleaned and filtered data. This can then actually have two outcomes. So it might be that the building of the model is the interesting thing, and we can report that. So if we're just interested to see whether the variables that we've included can predict our outcome, that might be interesting to us. And so if we can just build a good model, that might be all that we're interested in, and we can report that. But usually what we want to do with machine learning is make some kind of prediction. So we want to be able to create a model off of some data that we've collected, and then use it on future data where we maybe don't know the outcome, and we can predict based on that. So what are the general steps within this box here, the modeling step? Well, I say within this step. The first step, actually, I've still included is generating the data. This is because, again, it's super important. You want to generate good data when you are hoping to build a machine learning model. We've all heard of rubbish in, rubbish out. That is very true. So you want to produce as large data sets as you can, ideally, and at as high quality as you can. And this can often be a challenge, but is necessary to build a good model in the end. We then have to pick a modeling method and build a model. And I'll come back to how we might decide on what model we're going to use, but that will depend on a few features of our data that we've generated, and also on what outcome we want to get, as we'll see in a bit. Once we've decided on our model and built it, we'll want to evaluate it, so see if it's any good or not, and this will likely repeat a few times as well. So it might be that you try one modeling method, evaluate it, it's not very good, so you try a different one. You make some tweaks and changes to some of the parameters and things like that. And eventually you'll get a final model that you're happy with, and you can make predictions where the outcome is not yet known. And so once we're able to do this, we can also then test the predictions on new data that we haven't seen before when we were building and making our model. So that's the sort of overall steps that we follow at a sort of conceptual level. As I said, the model type that we want to predict will depend on a few things. The main differences between models are the outcome type that we want and the input that we have, or the type of data that we have. So in terms of outcome type, there are two main types of models. There are regression models, which will make quantitative predictions. So for example, if you want to predict height in your samples, you will want some kind of numerical quantitative value, and therefore you will want a regression-based model. The other outcome type is a classification model, which is for categorical predictions. So if, for example, you want to classify a sample as healthy or not, that will be classification because you're just saying yes or no. Classification can also be more than two groups. It doesn't have to be yes or no. It can be however many categories you have. Some model types can do both, as we'll see in a bit, but if you know you definitely want a regression model, obviously that's going to restrict you to models that do regression, and if you only want classification, that's going to restrict on those. Sometimes you'll have a choice. So the other side is the input data, as I said. Some models required all of their variables to be numeric, so there are sometimes cases where you have to convert your categorical values into numbers. So for example, you can make yes and no 0 or 1. There are a whole load of different ways of converting to categorical values, but sometimes you do have to do that. Also, some models expect certain behavior of your input data. So some models, for example, require your input data to be normally distributed. And so that can also be a deciding factor on which model you use. And there's also variation in the number of viable measures that you can use. So, for example, some models might only need hundreds of data points, some might need thousands, some work well on large data sets, some don't scale very well. So, depending on the actual size of your data set that you have to train your model, might also influence your choice. So, some of the models that you might see referenced at different points, and some of these I'll go through in these seminars. All of these I go through pretty much in the machine learning course that I do, if anyone wants to learn more after this. But the one we're going to look at today is linear regression. The first one on here, as you would expect, is a regression model. Surprising from the name, I know. But some of the others, for example, like naive Bayes, decision tree, random forest, are more classification based, whereas some of the others can do both. So logistic regression, k nearest neighbors, SVMs, and neural nets. We will go through, as I say, some of these different ones at different points, but I'm not going to talk about any of these except for linear regression today. So on to the paper that we're going to sort of base the discussion today on. It's this paper here, titled DNA Methylation Age of Human Tissues and Cell Types. This is essentially a paper looking at epigenetic clocks. So what they're doing is trying to use DNA methylation patterns across a range of different tissues in this paper to try to predict biological age based on this. And they used over 80 different publicly available data sets in the paper. They had quite a large number of samples, nearly 8,000, across sort of about 50 different healthy tissues and cell types. Something to note, which is important when they do the pre-processing, is that across the different data sets, there were a couple of different arrays that were used. It doesn't really matter the details of this for the purpose of this seminar, but just something to be aware of, that they were dealing with slightly different or slight differences across some of the data sets. And because they had these different data sets, they did something slightly differently to how we'd perhaps usually think about machine learning. So with machine learning, you always have to have a training data set and a test data set that are separate from each other. So you train your model on one data set, and then you test it on data that it hasn't seen before to see how well it works. Often you have a single data set, so you split your data set at that point. But in this case, because they had these quite a large number of different data sets, they instead split the data sets themselves. So the first 39 data sets that they had were used to train the predictor. They set a few different criteria for this. So they wanted to have a wide spectrum of tissues and cell types in their training data so that they had good data across different tissues and cell types and hopefully could get a broad prediction across those. They also tried to keep the mean age and standard deviation similar to the test data. So they didn't want to train just on young people in the training data and then have all the older samples in the test data. They wanted it to be broadly the same and cover a sort of good range. So both of them had standard deviations of 25 years and a mean age of 43 in the training and 42 in the test, but pretty similar. They also tried to keep a relatively high proportion of, I believe, the newer of the Illumina platforms for this. So they had slightly higher proportions in the testing data in the training data rather than the testing data. They then had a whole load of data sets that they used to test, which I sort of discussed up here. And they had about 10 data sets that they used for other things. So after they'd trained and tested their data, they then sort of put it to the test in more unusual situations, which is where these data sets came in. So they had data on things like embryonic stem cells and IPS, or induced pluripotent stem cells, to see how their model looked in those kind of edge situations. So that's the data they had and how they split it up. Now we're going to focus on the training data predominantly, although the same process was also applied to the testing data. So this is the pre-processing that they did in there, or before they ran their model. So they had just over 21,000 CPGs, and they had then methylation data for each of these, which measured on across these two different Illumina platforms, as I mentioned. So they filtered out CPGs so that they were only including ones that were present in both platforms, and they also filtered out ones that had more than 10 missing values across the data set, so they had pretty good complete data overall. They then normalized all of these so that they were essentially rescaling so that all of the different data sets had the same distribution. They said they adapted this BMIQ function, but I'm not going to go into details on that because I do not have time. But essentially they've normalized it so that all of the data sets have similar distribution and can be more directly included in the same data set. The final thing they did, which is probably the most interesting on this page, is they transformed biological age. So if you can see on the top right up here, these plots show age versus the sort of average methylation values within the data set of the training data on the left and the test data on the right. And you can see before the age of roughly 20, there's a sort of log linear relationship between age and methylation. And then after that point, it looks fairly linear. So what they did is they transformed biological age based on this cutoff of about 20 years, saying that anyone over is 20 is considered an adult, and then they have a sort of linear relationship, and below that point, there's this more log relationship. So essentially all this does is log transform those who are less than 20 and linearly transforms those that are over 20.
So what we're doing.
I think someone's unmuted, so I'm just going to mute them, assuming that wasn't a question. So this is just an example on the right here. You can see this is linear age as was originally, and you have this sort of log relationship here. It's harder to see because there's fewer data points, but then when you transform the age, which generally is called F age like this, you get a much more linear relationship between age and methylation. This is important because we're going to do a linear model that expects a linear relationship. So this allows them to then use that model later on. So the model, or the input to the model that we have, therefore, is our normalized methylation values from our 21,000 filtered CPGs. The output that we want is to predict the transformed age, because we're going to use the transformed age to train it. And so therefore what we're going to want is some kind of regression model, because the transformed age is going to be some numerical value rather than a categorical value. And we know we have linear relationships now, so we can use a regression model that has linear relationships between our variables. So the model that they used in the paper is called the elastic net regression. I've put the linear in brackets because it is a linear regression, but usually it's just called elastic net. So to explain what this is, I thought I'd do a quick overview of what normal linear regression is. I'm sure many of you are well aware of this, but just as a reminder, linear regression generally depends on or models the dependence between our two variables. So we'll have an X variable that is our predictor and our Y value that we are trying to predict, and it will draw a line of best fit through our data. Because we have a line of best fit, we will have an equation associated with that, and that will form our model. So our Y will be equal to two coefficients that are calculated as part of the model. So beta naught, which will be the intercept of the line, and beta 1, which is the slope. As we go through this, I will refer to this more as the coefficients than the slopes, but that's sort of a nice way of visualizing it internally, I think. And so this means that we can use our model. If we have a new value of X, we can plug it into here. If we know our line of best fit, then we can calculate a predicted value for Y. And we can expand on this to create a more complicated linear model by adding in extra predictors. And we can literally do this by just adding in an extra coefficient and then timesing that by our new predictor and adding it to our previous predictor. And this can be expanded as many times as we wish until we have sort of n number of predictors, however many we want. And we'll get an equation that looks something like this. And I promise this is as complicated as the equations will get. But essentially, we're just weighting each of our predictors here by some coefficient that essentially says how important that predictor is for calculating our value of Y. So if it's not very important, that coefficient is likely to be relatively small. If it's really important, it might be larger and have a larger impact on Y. And that can be either positive or negative. Normal linear regression is actually what's termed least squares regression. And this essentially aims to minimize the sum of squares on the model. And the sum of squared is just the sum of our residuals. So as a reminder, the residuals is essentially the difference between our actual data point and what the model thinks our data point should be. So for this data point, the residual is here. It's the difference between our point and our model, which is in this case our line. To get our sum of squared errors, we take this residual, square it, add it to this residual squared, etc., etc. We square it because it means that then they're all positive, but it also then weights the larger distances. So larger distances get squared to be even larger and smaller distances don't really add that much. So sum of squared errors is a nice way of measuring how good the model is. But also when you minimize that on a line, you will always end up with the same solution. So if you fit a line of best fit on here, it will always be this line. You can imagine if you tried to fit a horizontal line, you would have much larger least squares because this point would have massive residuals upwards to this point, and so it would then move it to average out to be the same slope as it is here. This is great, but it only works when our number of predictors is greater than our sample size. And so to help visualize that, I've got some again, just 2D examples because they are much easier to visualize. So if our number of predictors is less than our sample size, we can't really solve for our least squares, or equals our sample size, really, I should say here, sorry. So our number of predictors here is just 1, and then we're trying to predict Y, so we only have one data point here. And clearly we can't fit a line with least squares because we could do this line, we could do this line, we could do any of these lines, and they're all going to end up exactly the same because there's no error whatever you do because you only have one data point. If, on the other hand, you only have 2 data points, you can then fit a line, but it's going to be perfect for these 2 data points, but probably not great once we add in extra data. So the chances of extra data falling exactly along this line is probably quite slim. If you had in more data points, you'll get a much better prediction for your line of best fit. So how can we then cope when we're doing machine learning models where we have a large number of predictors and a relatively smaller sample size. So we had, even if we have like 8,000 samples, like they did in the sort of entirety of this paper, when we have 21,000 methylation values, that's quite a lot smaller. So what we can do is something called regularization. And this allows you to perform a regression when your number of predictors is greater than your sample size. It also reduces overfitting when you just have relatively few data points compared to your variables. So it's good generally when they're sort of on the edge of being the same. It does this by minimizing the coefficients. So essentially minimizing the slope, which minimizes the impact of a given variable. So if we see this slope here, if we add some regularization, what we're essentially doing is adding a penalty to the model. So we're going to make the slope less steep. And this means that for each increase in x, so if we go from 1 to 2 on this original line, y will increase from 2 to 3, but on the sort of less steep slope now, it'll only increase by sort of 0.5-ish. So the model is now much less sensitive to increases in y, and they will have less of an impact on the end value. Of increases in x, and they'll have less of an impact on the final predicted value for y. And how much of a difference this makes depends on how much of a penalty you add to your model. So if you increase the penalty, you will decrease the slope or the coefficient, and you will reduce the impact of that particular variable on your model. So that's the sort of overview of how regularization works. There are actually several types of regularization that you can do. So you can do ridge, lasso, or elastic nets are the main ones you'll hear about. All of these essentially have the same idea. They all decrease the sensitivity to your variables. They reduce overfitting overall, and they allow you to have more predictors compared to your sample size. The first one, ridge regression, adds a squared penalty. I'm not going to go into the maths behind this, but essentially if you add a squared penalty, it will mean that your slopes or coefficients tend towards 0 when you increase the penalty, but will never actually reach it. So this is good when all of the predictors that you're including in your model are likely to be important. So if you want to keep everything in your model and you think that all will be useful for predicting your y, then you could use ridge and it will add a particular penalty, but it'll never go down to 0. Lasso, on the other hand, adds an absolute penalty. Again, I'm not going to go into details on that, but it basically means that your slope or your coefficients can be reduced to 0. So this is good when some of your predictors are likely to be less good, because it means that they can be removed. So you end up with a much simpler model at the end. Elastic net is even better because it combines both ridge and lasso regression. So it adds both squared and absolute penalties, which means that you can still remove poor predictors, but it's a bit less aggressive than lasso tends to be. So you'll still end up with the majority of your predictors and likely the important ones. It's particularly good when you're not really sure how good your predictors are, but on the whole, people would tend to go to elastic net because it sort of ends up being the best of both for both ridge and lasso. So given all of this, we're going to have to give a particular penalty value to our model when we make it. That sounds intimidating. But we can estimate our penalties using something called cross-validation. We would have to do this whether we use ridge, lasso, or elastic net. So they did have to do this in the paper. And cross-validation is really important and a useful concept in machine learning. So I'm just going to very quickly go over what it actually means. So as I said with machine learning, we're going to need some kind of training data set and some kind of testing data set. so that we can train our model and then evaluate how well it's doing. Cross-validation allows you to do that multiple times, essentially. So if we start off with this training data here, we can split it up into 10 different proportions of our data. And we could train a model on 90% of our data and then test it on 10%. We could then do it again, but take a different section of our data and then again and again and again until every 10% portion of our data has been trained in 9 models and tested in 1. In this case, this is tenfold cross-validation, which is the most common one. You can do it different numbers of times, but 10 is often used. And this is really useful, particularly when you're trying to figure out parameters to use in your model. So if we're not sure what penalty value to use, we can try different values and see which the best one might be by using this cross-validation. And that lets us optimize the parameters that we're using in our model, essentially. So that's exactly what they did in this paper. This is the model parameters that they used when they built and trained their model. It looks like this here. We are not going to worry about this because I'm going to show you some much nicer code later on for doing this. But essentially the headline is they used this function called glmnet, or this package, to train their regression model. So this is just a function in R that will build and train this particular model type. However, it does require 2 parameters. In this case, they're called alpha and lambda. Alpha just defines the regularization type that we want to use. So for an elastic net, it will be 0.5, and then it's 0 and 1 for either ridge or lasso. Lambda then defines the level of penalty applied. So this is what they optimized using a tenfold cross-validation on their training data, and then they ended up with this value of 0.0226. We will come back to how we would look at running this model ourselves, but just to show you how they have done it here. So then when they did that, they got an output that was a linear regression model with a number of coefficients, actually beta 0 because there would have been an intercept, but then beta 1 to beta n for all of their included CPGs. So for the 21,000 odd CPGs that they included, they would have got a coefficient for all of them. However, many of these were removed and had a coefficient of 0, which essentially just removes them from the model. And so they ended up with 535 CPGs in the final model, and so in their final epigenetic clock, as it's termed. The coefficients that they got related to the transformed age, because that is what we got at the input. Again, we'll come back to visualizing this more in a minute. But essentially, using their model, you could calculate the sort of chronological age or the actual age of the person by inputting all of the coefficients that they got from their model and timesing it by each of the related CPG methylation values. And then there will be some kind of error associated with that. You can then use the predicted age or you can inverse the transformation on the predicted age to get the actual age on the same scale as the original age, which is much more useful to actually conceptualize. So they built this function here called inverse f, which again, we will see a bit later. Yes, sorry. There's a typo on this slide. You're absolutely right. This should be 353 CPGs, not 535. That's a good spot. But yes, you're absolutely right. Yes, I wrote it right down here, look. So yeah, 353 CPGs were included, not 535. So yes, they ran this model, they got all of these coefficients, and now they could evaluate it. And apologies for the very small image. I could not find somewhere that they'd put a higher quality one. But this shows the results from the test data. So they evaluated their model both in the training data and in the test data. As you would expect, the training data behaved slightly better than the test data because it had already seen it before. It was used in the training. So in this case, age correlation relates to literally the correlation between predicted age and the actual age of the sample, just using Pearson's correlation. The median error is the median absolute difference between the predicted age and the chronological age. And they refer to their predicted age as DNA-M. So I've used that as well. So essentially in their test data, they had a pretty good correlation of 0.96. That's really good. And their median error was 3.6 years. So essentially that means, as you can see here, that the predicted age differs by less than 3.6 years in around 50% of the subjects. So again, pretty good, especially because in the test data, this is looking across a whole load of tissues. They also looked at individual tissues though. So the rest of the plots are looking at individual data sets or tissue types or individual cell types as well, and using their model on those to see how well they perform. On the whole, it was pretty good across the board. A lot of the correlations were sort of 0.9 plus, certainly 0.8 plus. There were a few where it didn't look to work so well, but it's also worth noting, and this is something that's always worth considering when you're evaluating your model, is how good your evaluation metrics are. So the correlation between the two did depend on the age range within the data set that it was tested on, as you might expect. So for example, this first row here in the CD4 T cells, they had a really small age range. They were all really young samples. And so the correlation is perhaps slightly lower because of that. And in this row here, you can see the adipose tissue only ranges from like 73 to 78, which again is quite a narrow range, and the correlation there is generally lower. And they did plot this out and show that it is actually correlated as well. But on the whole, looks to work reasonably well across a whole load of tissue types and different cell types, across a range of age ranges as well. So now we've seen how they've done it. I thought it would be useful to do a short practical demonstration of how we might do it in Tidymodels. So Tidymodels is essentially, it's sort of built within the tidyverse of R, and it's meant to be a consistent interface to deal with machine learning problems, essentially. So it means that instead of having to use a load of different packages for different model types, you can just use Tidymodels and it will essentially translate different packages that have built the different models and let you use them in a much more user-friendly way. I don't have loads of time to go into loads of details on this, but if you want more details, you can go on their website or find their book or come along to the machine learning course in July. But essentially some key things to say quickly about this is Tidymodels works in a slightly different way to how you might usually expect when coding in R. So what we do when using Tidymodels is create essentially an empty model first. And this is the sort of slightly weird thing, but it makes sense and is nice to use once you're used to it. So you create a model with no data in it, just the type of the model, and you can set the parameters and things that you want to use. Then you sort of separately create your data. So filter it and prepare it in whatever way you need to. So do your filtering and scaling and normalization in whatever way, extract any features, things like that, and then split off training and testing data and set up any cross-validation that you might wish. Then you combine your model and your data. to train your model. So you pass your data into the model and define the variable that you want to predict at this point too. And then you can use your trained model to test and use the model on new data types. So to do an elastic net in Tidymodels, we're actually using the exact same package as they used in the paper, GLMnet, but we're using it in a slightly nicer way that is more similar to using other models within Tidymodels, rather than having to learn a load of different packages. So within Tidymodels, we still have two parameters that we need to give our model. These are very familiar though. So the first one is the penalty, and this is the amount of regularization, the amount of penalty that we want to add to our model. In GLMnet, this was called lambda. And the second one is called mixture. This was called alpha in the GLMnet, but this is the proportion of the penalty, of lasso penalty, sorry, is what it's called. So essentially it goes from 1 being a pure lasso model to 0 being a pure ridge regression model. To get a complete mix of the two, elastic net model, then you can use 0.5. I believe you can use things in between 0 and 1 that are not 0.5 for different weightings on lasso versus ridge, but we're not going to worry about that. We're just going to go with the complete mix of the two at 0.5. Tidymodels also gives us some pre-processing requirements that we would need for this type of model. So we need to have numeric values as the input. So if we had any factor or categorical predictors, we'd need to convert those. That isn't an issue here because they're all numeric. They're all the methylation values. And the predictors should all have the same scale. Again, they did this in the paper when they normalized things and also made it linear using the transformation of the age. So the actual code that we would need to build our empty model up here is this linear reg command. Again, we're setting the penalty, so I've pulled the penalty from the paper in this case, and our mixture is 0.5. We then use this set engine command to tell it that we want to use GLMnet, and then we can save that into our linear regression model. And then we can take our linear regression model and feed in our data using this fit command here. So in this case, I've just shown some input data, and the formula that we use here tells it what we want to predict. So we want to predict F age from, in this case, the dot means everything else. Okay, so we can now have a look in R. So here's some code I made earlier. We'll run through it and see what it does. So the first thing we need to do is load in the libraries that we want to use. So we want to use Tidyverse and Tidymodels, obviously. We can also use this here called Tidymodels prefer. That just means if there's any overlapping sort of functions with anything else, it will select Tidymodels ones preferably. Again, don't really need to worry about that too much at this point. And this theme set is just for aesthetics, essentially, of graphs. So we're going to run that, and that will just load those in. Then we can build our model. So this is exactly the same as I showed you on the slide here. So we're using our linear regression model. We've inputted our penalty from the paper. We've set that it's an elastic net model, and we've set the engine. So if we run that, and we can also have a look at it, we can see it tells us that this is a linear regression model specification. It tells us the arguments we've given it, and it tells us that we're going to use GLMnet. The other thing you can do in Tidymodels is use this translate, and it will show you what it's actually giving to the GLMnet package. So if we run that, it again gives us our main arguments and our engine here, but it will also show us the GLMnet function that we have down here. You can see there's a load of missing arguments. This is because this is an empty model currently, but it's inputted our alpha here too. So we've got our model set up. now we can read in our data. So this is some I prepared earlier. So I'm just going to run this and we can have a look at it. So you can see we've got sample IDs on the left. That's just our different CPGs. We've got the actual transformed age of our samples. No, sorry, these are the sample IDs, not the CPGs. The CPGs are across all of the rows with the methylation data here. These are the sample IDs of the actual samples. And then the transformed age that we have here. So to input this into our model, we actually don't want these sample IDs because they're just a unique identifier. They're not going to be of interest. So the first thing we can do is just remove this column before we input it to our model. So we can do this using the select command and then just remove that column there. And we can see now we get the exact same data, but just with the age and the methylation there. Now we can fit the model. We can use set.seed. This just essentially controls the randomization, so you'll get the same result every time. And then we can, as I showed again in the slide, we take our model, we use this fit command, and then we give it our f age, which is what we want to predict, and we'll give it our input data there. We can run that. With these linear regressions, you can just look at this fit here, but it doesn't really tell you that much useful. So you get a much nicer output if you use this tidy command here within the Broom package, and that will let us pull out our weighted coefficients. So if I run that, you'll see we end up with a load of different coefficients. These are for each of our CPGs. I should have said earlier, I filtered this data set so it is not a big one. That's why everything's running super quickly. And we're also not going to end up with a very good model at the end, but it's just useful as a visualization. Because of this, I also filtered to just include the 353 CPGs that were included in the clock model. So I filtered out all of the ones that were filtered out in that model. That's why we have 354 coefficients here, as you can see from the number of rows. So the 353 CPGs plus an extra row for the intercept term here. The first thing you can notice here is that most of these are zeros. So most of these have not been deemed to be important in this model. Again, it's a much smaller data set. There's not that many data points. So that's not surprising necessarily that they're not interesting in this particular case. But what we're going to do is remove all the zeros so we can just find the interesting ones. So if we take our coefficients, we can filter it just to include the non-zero ones and also remove the intercept because that's not super useful to us at this particular point. And so we can see that there are 32 different CPGs that have been included in our model in this instance. And we can see some of these are positive and some of them are negative. So we can count how many there are and see that the majority of these are negative. So they'll decrease with increasing age rather than increase with increasing age. The next thing we can do then is see how well this works on new data. In this case, because it's an example, I'm just reusing the same data, so it will not be a real evaluation of the model. Clearly, this is not what you would do, but for the sake of simplicity, we're just going to rerun it on the same input data, and then we can use that to see the sort of process generally. So to do this, we can use this predict here. We'll give it our linear regression fit, so this is the trained model. And again, we're giving it our input data. Usually this would be your testing data rather than your training data again. So if we run just this bit here, you can see this just gives us a column with a load of values. It's not super useful. That's our predicted age, but it doesn't really mean a lot when you don't know what the actual age was. So the next thing we're going to do is use this bind calls. So we're just going to join it to our input data And then I'm just selecting the predicted age and the actual age here, just to make it more neat. So if we run all of that, we can see we have a predicted age and an actual age of the sample. This is, however, the transformed age, because that's what we used as the input. So we can inverse this transformation to get something that is more useful to us. So from the paper, they wrote this little function which just converts it back to normal age. So we're not going to worry about that too much, but we will use it anyway. So if I run this, we can see now we have our predicted and actual transformed age and the original age scale on these two columns here. And so we can just plot those against each other and see how well it's done. As we would expect, this has worked beautifully because we have so few data points. It's not really a very good example of building a model, and we're also giving it our trained data back. But that's how you can do it in theory. And we could also look at calculating the correlation between these two, which you would expect to be super high. For stats things, usually I use this rstatics package. So I'm just going to load that in, and then we can use this core test here, and that will run our correlation. So the correlation coefficient in this case is so close to 1 that it's just been rounded up to 1. So super high, as we would expect, given how we've done this example. So hopefully that gives you a brief overview of how we might go about doing this ourselves. I thought the other thing that's interesting to have a look at this point, which I'm not going to run through in R again for the sake of time, but is how changing our different model parameters affects the final model that we get at the end. So this one on the left is what we just did. We used the penalty from the paper. We put our mixture at 0.5 to give us an elastic net model. And this included 32 CPGs in the model, 5 of which were positive, 27 were negative, and it looked like this. If we increase the penalty, then we reduce the slope. So we end up with essentially a worse fit for our data that we have in our training data. So clearly this line, or the predicted, is not as good in this than this, but it might be that if we then have more data included in our test data, it might fit it better because it's potentially less overfit on our original data set. We ended up with fewer CPGs included in the model tier 2 because we had a higher penalty, so more were removed. We could also alter it so we only use bridge or we only use lasso regression rather than using elastic net. I kept the penalty higher here because it's easier to see. So for ridge regression, we put mixture at 0. In this case, we have all 353 CPGs included in the model because it uses the squared penalty, so it will keep everything. And this, in this case, ends up with a better model than the elastic net here. Again, as we might expect, we don't have a whole load of data going into it anyway. And also we filtered our CPGs to include ones that we know in a much larger data set have been shown to be useful. So that kind of makes sense. And we can also change it to be lasso. In this case, it filters out basically everything, so we only have one CPG included in the model, so it looks much worse in this case. Again, that won't always be the case across all datas. This isn't, or across all data sets, this isn't saying that lasso is bad and ridge is good. It's just for the particular situation of this example, that's how it looks. Okay, so the final bit, I thought it's just useful to think about how they then used their results from their model to sort of further explore or explain what they've come up with. So the first thing I think that's interesting when you're doing a model like this is obviously what is contributing to the model. So when you're doing a linear regression, you get the coefficients for each of the CPGs, so you can pull out the interesting ones and see what they are. So they looked at doing pathway analysis of the genes that co-located with the clock CPGs and found enrichment for like cell death, survival, cell growth and proliferation, and development, also cancer. If you're familiar with the paper, you'll know they did a whole load of stuff with cancer data sets too, which I've completely ignored for simplicity, but they did also have a look in more depth at that as well. They also had a look at how the predicted age looked versus actual age in chimpanzees and found that that correlated pretty well, actually. So that's quite interesting. I think they also looked at gorillas and they didn't correlate quite so well, which sort of makes sense. And then they also had a look at embryonic stem cells, which had a predicted age close to 0, which is nice. And also predicted age in iPS cells, so induced pluripotent stem cells, was younger than that of the corresponding primary cells, but was no difference to embryonic stem cells. And finally, they found that cell passaging or cell splitting, so growing your cells in cell culture, increases the predicted age too. So that's all quite nice, I think, and shows how you can take your model and apply it to different data sets and see how it works within those that might tell you interesting things, either about the things that you're trying to predict or about your model itself, and see how well it's doing. So that was a sort of brief overview of that paper and linear regression. Hopefully you find it helpful and interesting, and if so, there are 4 more of these sessions that you can come along to. As I said, these will be looking at different model types and different input data types, to hopefully give a broad idea of what you can do and how people have used machine learning in sort of relevant areas already. So hopefully, because you're all here, you've already found this, but this is just to say there's the Zoom link via this QR code. And also I will be doing an introduction to machine learning course on, I believe, the 14th of July.
Yes,
But you can find,
14th.
sorry.
Yes, it's the 14th.
Thank you. But you can find the course dates via this QR code as well and sign up there. So if you are interested in machine learning and want to know more, please feel free to join that. Otherwise, thank you for listening.